Good morning, everyone. My name is Ann Wolfe, and as Manager of Strategic Advancement at Global Genes, it is my distinct pleasure to welcome all of you here today to our 7th Annual Rare Drug Development Symposium. On behalf of myself, our Global Genes team, the organizers, and the Orphan Disease Center at the University of Pennsylvania, we are so excited to have all of you here today, and thank you all so much for everyone who has taken time out of their busy days to spend this day with us. We hope that this is a great experience for all of you. Now, I'm not the feature today, so I will be turning this over shortly, but I wanted to take care of just covering a few housekeeping and important logistical remarks. So, first, you will see that the chat is available to the side of your video screen. The chat allows you to interact with each other and ask questions of your peers. However, we do recommend that you use the questions box to actually ask the questions to the presenters, as they are not able to see the audience chat. If you'd like to share any resources, we recommend that you share those on the social wall rather than in the chat, as the chat does not allow for links or other documents. We encourage you to use this time to ask all of your questions, and we will do our best to address as many as possible. However, it is likely that we may not be able to get to all of those, so if we are not, we will be sure to take those offline and do our best to follow up with everyone after. You will also see to the right of your screen the option for captions and translations. To utilize either of these options, please expand that section of your screen, and then you have an option to choose from a number of languages. Additionally, you are welcome to utilize the notes feature to the side of your screen as well. If you utilize that feature, your notes will be emailed to you directly after this session, which is a great way to keep track of your notes throughout the program. If you have any questions of technology throughout the program, in session, you can see the request support button. And when you are outside of a session, you can see the live support chat button. Either of those options will connect you to the production and tech team, and they are more than happy to assist you in troubleshooting any issues. And finally, we just want to encourage everyone to participate as active listeners. We know it's easy to get distracted in this Zoom world, and we want everyone to really enjoy the program, take time to listen and absorb this knowledge, and hopefully learn something and find things to take back to your community. We're so excited again to have all of you here today, and thank you to everyone who has contributed to making this program a success. We encourage you to share your feedback with us throughout the program, as we always want to continue learning how we can better provide programming. And so, without any further ado, it is my pleasure to turn this now over to Craig Martin, CEO of Global Genes, who will share our official opening remarks for the day. Craig?
Thank you, Ann. I'm pleased to welcome all of you to the 2021 Rare Drug Development Symposium in partnership with the Orphan Disease Center at UPenn. I want to offer some special thanks to our panelists and speakers for contributing their time and expertise to this event, as well as the companies and advisors that you see listed here for their support and guidance. And although my comments are technically billed as the opening remarks, I do want to mention the pre-conference workshop that was held yesterday. It was really well attended and very engaging. And the level of discussion that took place, I think, was a testament to our co-chairs and to our speakers, as well as to the desire of those involved to really learn more and get the most they could out of this event. This is the 7th year that we've held this event with our partners at ODC, and David, whom you'll hear from in a moment, was a driving force in getting this going. Clearly, there was and is a need. Each year we've grown, even last year, and had an opportunity to reach more rare disease community leaders, researchers, industry representatives, and others. And in just a few minutes, you'll hear a little bit more about the Orphan Disease Center and their great work. But I wanted to share a little bit about Global Genes as well, in part so you have some context as to why this partnership and the topics that we'll discuss the next couple of days are so important. So Global Genes is a nonprofit dedicated to driving progress and addressing needs across the more than 8,000 rare diseases affecting more than 300 million patients worldwide. Our mission is simple: eliminate the burdens of rare diseases for patients and families around the world. The path to get there is not easy. with so much unmet need and more rare diseases being defined all the time, we focus on enabling and empowering rare patient communities through events like this one to organize, mobilize, connect, and be better equipped to drive progress in their disease areas, as well as to work together across rare diseases and stakeholders to address challenges that stand in their way. Our ultimate goals are to dramatically shorten and improve the diagnostic odyssey that is so devastating to families, patients, and clinicians, and to speed innovation and improve access to treatments and cures. In pursuit of our goals, we work with individuals at the earliest stage of their journey, sometimes even before diagnosis, through our Rare Concierge service. And we look to direct patients and caregivers to a next step that will give them forward progress, which could be genetic testing or access to a geneticist, or connecting them to a community or to a trial, or even to an event or some educational content. As we look to scale up our services, we're also developing the technology and tools to enable patients and caregivers to collect and carry information and experiential data along with them, and to manage and inform their interactions within and beyond care, ultimately then to aggregate and share their experiences and insights back to their community. From there, we help communities to form and work together and build their capacity to generate interest and drive progress in their disease area, helping them to get to a point where a researcher or a company may take interest in drug discovery and development in their disease area. And then we work with established groups as part of our Foundation Alliance, which now includes nearly 800 rare disease patient groups, helping them to continue to advance progress in their disease and to work together on critical issues and needs, and to provide venues where they can give back and help other advocates, patients, or caregivers who are earlier on in their journey. Our support and involvement can come to life in events like this one, which provide an opportunity to not only educate and equip patient communities to participate in drug development, but also to connect them to others who can help and guide them along the way. In other instances, we may source and provide impact grants, like the Health Equity Impact Grants we just awarded, which are designed to build cultural competency and inclusiveness within and across rare disease communities. Or we work on initiatives like our Rare Compassion Program, which pairs medical students and someday soon, we hope, young clinical investigators with rare disease families to increase mutual awareness and understanding and encourage future clinicians and researchers to pursue a career specialized in rare disease. And as of this year, with the launch of our Rare Community Portal, we have created a place where the conversations, the connectivity, content, collaboration that begins with events like this one can continue in online communities that establish and grow over time from event to event, and bring the learnings from those community interactions back into the community to guide efforts moving forward. As we approach today and tomorrow and the months ahead, we have to acknowledge the ways in which the world has changed. In some cases, accelerated by COVID, but in others, just by advancing science and technology. Virtual has become our new reality, as we're all experiencing today. While that makes meetings like this one maybe a little more impersonal, it also allows us to reach more people. And when thought of in the context of virtual trials and telemedicine, it opens up other possibilities for us to make a bigger impact and level the playing field in research and care globally. COVID also further exposed the depth of structural inequities within the healthcare and research ecosystems and the degree to which those health inequities influence outcomes. It also brought to life, as we examined issues like diversity of odysseys, how little we truly understand about how those inequities manifest themselves in rare disease diagnosis, trials, and access to care. We, in fact, have an overabundance of underserved in rare disease. And not just based on race, gender, geographic, socioeconomic conditions, but also on population size. OrphanNet figures suggest that about 390 rare conditions are tied to as much as 98% of the rare disease population. That means that the remaining 2% of patients are spread across thousands of other ultra-rare conditions. how do we get drug development going in those disease areas? One answer may lie in platform science and technologies, and there's great reason for optimism there. The advancements in gene therapy and gene editing coming out of places like the Orphan Disease Center are astounding. And many of the more than 1,000 regenerative medicines in clinical development around the world are focused on rare disease indications. Importantly, not only are we seeing tremendous advancements in technology and discovery for rare disease, we're also seeing new collaborative and creative drug development and financing models emerging that may help to drive more innovation out towards underserved populations and ultra-rare diseases. We're in fact moving beyond traditional nonprofit funding of research and even venture philanthropy and into what I'd call philanthropreneurialism. That's probably too much of a mouthful to catch on, but the point is that patient communities are becoming more sophisticated in their understanding of what it takes to drive a therapeutic innovation forward. They are building their own companies to move these innovations ahead, and they're going much further downstream in therapeutic development than in the past. The Institute for Life-Changing Medicines, Columbus Children's Foundation, disease-specific patient groups, and even venture firms themselves are looking to change the way we approach discovery and development in order to make meaningful therapeutic advancements more accessible and affordable and get them to market faster. These models all include patient communities as partners and seek to involve them much further upstream in the process. In addition, patients are recognizing that their data really is their data. And that data is a vital asset and point of leverage for them to use to build relationships, attract investment, sustain and grow their efforts to improve diagnosis and drive research and development in their disease. New nonprofit, collaboratively funded and developed platforms like RareX are emerging to help them do just that. All of these shifts and developments point towards a promising future and towards some really interesting discussions over the next couple of days, and I can't wait for that to start. So with that in mind, I'm now pleased to introduce Cara Weissman of the Orphan Disease Center to share a bit more information on ODC and then to introduce you to our keynote speaker, Dr. David Faigenbaum. Thank you. Cara?
Thank you, Craig, for this introduction and for outlining the relationship between Global Genes and the ODC. As you had stated, my name is Cara Weissman. I'm the Director for the Program of Excellence for MPS Diseases at the nonprofit organization, the Orphan Disease Center, or ODC. Today, I will outline the way that the ODC works with patient groups to advise the development and support of research for rare disease in what we're calling patient-driven research partnerships. The origin of the ODC is a story that many here can relate to. In 2011, a Penn alumni found his family on the rare disease journey and searched for existing support, treatment, and research. He discovered this path was very challenging to navigate and realized that many families were facing similar struggles. He then engaged the researchers at Penn and through a philanthropic donation began the ODC, and then began our mission. The Orphan Disease Center will develop transformative therapies using platform technologies that can be deployed across multiple rare diseases. We will emphasize disorders with substantial unmet need, independent of their incidence, and will strive to assure the access to patients of all populations. We believe that being too rare or too poor should not preclude you from access to curative therapies. The ODC is a small but mighty team of 11 dedicated professionals taking on all the programs I will outline in this talk and many efforts behind the scenes. In 2015, Dr. Jim Wilson and Monique Malloy took over the leadership of the ODC and have crafted it to what it is today. Our name refers to orphan or rare diseases, which have had historically minimal therapeutic research associated with them. However, as this audience is aware, taken together, rare disease affects 1 in 10 Americans, or 33 million people. But this is a worldwide challenge requiring global solutions. Therefore, the ODC has a global reach, both in funding research and engaging scientists and patient groups. Our approach to this rare disease translational research is highlighted in these 4 areas, where many of our programs overlap to leverage research advances and platforms for multiple rare diseases. Often, one step to understanding what gaps of knowledge exist for a disease is to convene a scientific symposium or think tank. Here, we can ask the questions, is the disease pathology understood? What is the underlying biology? Current preclinical research and current treatments? What questions do we need to answer next? And what should be answered next? Most importantly, through both formal and informal patient advisory boards and meetings, what do the patients need? What will most improve their quality of life? Once a disease program has a direction, the ODC's pilot grant programs are aimed to address these gaps in research and knowledge in areas ranging from basic biology to animal models and through therapeutic development. These programs encourage the participation of new investigators with funding periods of 1 to 2 years and a range of funding from $40,000 to $150,000 a year. The intention is that pilot funding is a launch pad to more stable funding and additional therapeutic development. Importantly, in all of our grant programs, Million Dollar Bike Ride, Jumpstart, and the Programs of Excellence, the ODC manages all pre- and post-award activities, from the development of the request for applications through peer review, funds disbursement, research reporting, and the long-term outcomes from these programs. And of course, all research tools and results are shared. Since 2011, the ODC has funded 400 awards and more, totaling over $38 million to 120 plus institutions in 25 countries worldwide. Most of you are familiar with the Million Dollar Bike Ride, or MDBR program, our flagship fundraising event running for 8 years this weekend. Led by Samantha Charleston, rare disease teams and foundations ride as a part of a team and raise money for rare diseases to support pilot grant programs, as I mentioned earlier. Most importantly, the rare disease teams and foundations set research priorities outlined in the request for application, meaning you can influence the research you most want to see in your disease. To participate this year, you can support by donating to a team, riding in a virtual program or spin class, or see our webpage for additional details and participation in future events. Here are some of the amazing teams that will be with us again this year. The Jumpstart program is led by Debbie Reckerson and is intended for disease groups which either need a one-time consult to discuss the next steps in disease research or connect with external resources, or to become a Jumpstart partner for ongoing consultation and research development. Here's a brief overview of all of the areas where the Jumpstart program can support patient groups. From outreach to grants, Jumpstart brings together all of the ODC has to offer. For more information, please schedule a consultation with Debbie. Please see our website and information at the end of this presentation. And here are some of our Jumpstart partners. An established Orphan Disease Center Program of Excellence has a dedicated funding source with an internal ODC director leading the charge. These involve partnerships with rare disease foundations or multiple foundations and experts. These grant tools, programs, and resources are intended to advance the science towards therapeutic development and clinical trials, and to lower the barrier of entry for those additional funding to support those diseases. 2 of our internal programs of excellence are the Program of Excellence in CDKL5 Deficiency Disorder, or CDD, led by Dan Lavery, and the Program of Excellence in Mucopolysaccharidosis, or MPS, led by me, Cara Weissman. Here are some clinical trial highlights resulting from our internal POE grant programs, including 7 in CDKL5 and 4 in MPS. We then have 4 additional programs of excellence that are led by CHOP and Penn faculty directors, as shown here. We will get into greater detail about Dr. Faigenbaum, who has a unique approach and perspective in making tremendous progress for Castleman's disease. You'll hear more about that in just a minute. So how can patient groups work with the Orphan Disease Center? You can view or advise a scientific symposia. You can participate in the $1 Million Bike Ride. You can advise a request for application through MDBR. You can participate in a disease registry or natural history study, and the ODC supports quite a few of those. Engage in your National Disease Foundation or help to create one. However you choose to participate, please stay engaged with the ODC. We are here for you and with you to advance the science of rare diseases towards life-extending and improving treatments for all patients worldwide. Thank you very much for your time and attention. Please keep in contact with us for any of the outlined programs using the following websites and social media. And now, I am pleased to introduce my colleague and the keynote speaker today, Dr. David Faigenbaum. In addition to his role as an Associate Director of Patient Impact and leading the Program of Excellence in Castleman Disease, as the ODC, as I mentioned earlier, he's also the co-founder and president of the Castleman Disease Collaborative Network, Director of the Center for Cytochrome Storm Treatment and Laboratory at the University of Pennsylvania, a co-director of an FDA NIH CPATH public-private partnership called Cure Drug Repurposing Collaboratory, and a national best-selling author of Chasing My Cure, A Doctor's Race to Turn Hope into Action. And to David.
Thanks so much for that introduction, Cara. I am so excited to be with you for this session and to be with everyone joining us virtually to share about collaborations, community, and citizen science from lessons that I've learned from searching for treatments for COVID and also from my journey chasing my cure. So today I'm going to begin by sharing my story briefly as a bit of a background and then dig into a number of really key lessons from the effort that has been underway to identify drugs for COVID and also from my journey. I'll close with some future corrections. I did want to start out with one slide to try to share my personal journey and why I'm sitting here in front of all of you. I think it all started back around my first couple weeks of college when my mom was diagnosed with brain cancer. Her diagnosis with glioblastoma was just devastating for me and for my family, as I know so many people watching can understand and relate to personally. But her diagnosis and her passing a year later lit a fire in me to want to dedicate the rest of my life to searching for treatments for patients like my mom. And I promised her that I would dedicate my life to searching for drugs in her memory. And if you fast forward a few years to my third year of medical school, I was well on my way to hopefully being able to have a career where I could honor her memory through identifying life-saving therapies for patients like her. When out of nowhere, I became critically ill myself. I spent months hospitalized with multi-organ failure. My liver, my kidneys, my bone marrow, my heart, and my lungs all shut down. I gained 70 pounds of fluid due to the organ dysfunction, required daily transfusions and treatments and medical technology to keep me alive. I even had my last rites read to me when the doctors didn't think I would survive. Fortunately, I did eventually receive a diagnosis of a rare disease called idiopathic multicentric Castleman disease, a disease where my immune system becomes hyperactivated, it attacks, and then shuts down my body's vital organs. At the time, the only treatment option was chemotherapy, and chemo saved my life. But shortly thereafter, I learned of an experimental drug, the first orphan drug to ever undergo a randomized controlled trial for my disease. And I was so hopeful and thankful that there was finally a drug for me. I started receiving that drug, but unfortunately, I relapsed and nearly died shortly after beginning to receive that drug. And I realized that now the only drug in development for my disease wasn't working for me. And my only shot at survival and a dream of a future would be to get involved in research. And so I started conducting Castleman disease research at Penn. I also launched a foundation called the Castleman Disease Collaborative Network because I realized it wasn't something I could do on my own. I would need to connect with other physicians, researchers, and patients if I really wanted to make meaningful progress to save my life and hopefully to contribute to saving other patients' lives. Thanks to an amazing team and a lot of hard work, we eventually did discover a drug that had never been used before for Castleman disease, but had been around for decades, that I thought could maybe save my life. And I began testing it on myself. Thankfully, that drug is working, and I've been in remission for quite some time. I had the opportunity to share my journey chasing after my cure in a book, Chasing My Cure, which I have a photograph on the screen. If you haven't read it and you are in the rare disease world, I would love to send you a copy. Just send me an email, reach out to the organizers. I'd be happy to mail a copy to you. But thankfully and importantly, today marks 89.17 months since the last time I was in the hospital, and that I've been in remission on this drug. And you can see, compared to my previous remissions, this is about 9 times longer. But I say 89.17 months because I know that I can't round up. I don't know if I'm going to be in remission for 90 months on this treatment, but I also refuse to round down. It's not 89 months. I'm so thankful for every fraction of a week, a month, a day, a year that I've been able to receive thanks to this drug. And I've decided to dedicate my life to trying to find more solutions like this for more patients. So about a year ago, when COVID was emerging, and it became clear that patients with COVID-19 and particularly the most sick patients with COVID-19 experienced something called a cytokine storm, which is exactly what happens in Castleman disease, we decided we wanted to get involved. And so this is a picture on the screen of a Castleman's patient on a ventilator, on dialysis in the ICU, and on the right side is a COVID patient on a ventilator, on dialysis in the ICU. And if you look in the blood of COVID and idiopathic Maloof or Castleman disease patients, there's very similar immune hyperactivation. The only difference is that there's no evidence of a virus in IMCD. We don't know why the immune system goes out of control, whereas, of course, we understand in COVID. In addition to the similarities biologically between the two diseases, I also learned, and the world became aware, that a number of drugs initially developed for Castleman disease were actually being tried to treat COVID-19. A very famous one is a drug called tocilizumab. This is a picture of me with Kazu Yoshizaki, who actually discovered tocilizumab. I think I've shared with my Global Genes and ODC colleagues this story, but Kazu Yoshizaki made the discovery back in the early 90s that a particular molecule in the immune system called interleukin-6 was very important. He discovered this in patients. He developed a drug that blocks the receptor for that molecule. And then he was going to test the drug in Castleman disease patients. But before he did that, I had heard from my friend Makoto that Kazu had given the drug to himself. And so I went to Kazu. I said, Kazu, Makoto just told me that you gave tocilizumab to yourself as the first human to ever get tocilizumab. And he said, no, I didn't give it to myself. The nurse, she gave it to me. So Kazu discovered IL-6 was important. He created the drug. He tested it on himself. And when he didn't die, he went ahead and tested it in humans with Castleman's. He got approval in Japan for Castleman disease. It works in about 1/3 of patients. And then years later, it came to the United States and got approved for rheumatoid arthritis. And when the COVID-19 pandemic emerged, it began to be used for COVID. It also has been used in cancer immunotherapy. It's a picture of a young girl whose life was saved by it. And so I think it's so important to highlight these drugs like tocilizumab developed for a rare disease that has now had an outsized impact in very common diseases and in pandemics like COVID, which is almost the inverse of my experience, which is where there was a drug in this case, developed for a common disease, sirolimus or rapamycin for kidney transplantation, and now being used extensively in Castleman disease, a rare disease, and saving my life. So based on the similarities biologically and the treatments being used on Friday the 13th of March, 2020, which you probably all remember was the day the US shut down, was when our team ramped up. We decided that we would expand our Center for Cytocancer Treatment and Laboratory to focus on COVID. And we launched something called the Corona Project with an army of dozens. It's now been over 100 medical students from around the country that have dedicated countless hours to read over 29,000 published reports of patients with COVID and extracting data on all these patients to maintain what's now the world's largest database of treatments being used for COVID-19. And you can learn more about it at cdcn.org/corona. So as I thought about my talk for today, I thought I would hone in on 3 lessons from this journey that I think are really relevant for the broader rare disease community. So the first is that patient engagement is essential. We've all seen that we are starting to see the light at the end of the tunnel because of how many patients joined clinical trials of the COVID vaccine, participated in research, advocated for themselves and those that they love. So patient engagement is essential. This is something we in the rare disease community know well. The second is that all stakeholders must join together. It's not something that any one group can do. It requires multi-stakeholder collaboration and formal collaborations, not just we like one another, we work together, but truly formal collaborations where we're really leveraging the best and the brightest skill sets and perspectives and resources to get things done. And finally, drug repurposing is our fastest path to treatments. It's not our only path, and it's not even necessarily the best path, but it is certainly the fastest path. And as I say earlier, it's not the only path. We need to be thinking about definitive curative therapies, and that needs to be heavily invested in. But while we're investing heavily in definitive curative therapies, we also need to ask the question, how many drugs are already FDA approved that could somehow be repurposed that could help to alleviate suffering? So I'll start first with this, with patient engagement and a couple examples that I think are important. And actually, I'm going to share a brief overview of the framework we take through the Castleman Disease Collaborative Network as kind of, and then I'll zoom in to one specific example. So the way that we do work in the CDCN is a bit different from the traditional model for rare disease research. The traditional model involves raising money and then announcing a request for proposals where you invite the community to apply for your funding. And a small fraction of the community will apply for that funding. And then an even smaller fraction will be awarded the funding. And then research is done and progress is often made. This works really well for common diseases or diseases with lots and lots of researchers and money where you have a lot of people searching for solutions, but it doesn't work well if you've got 3 researchers in your entire field and 2 people apply for a grant. What is the chance that one of those 2 applications is going to be the best idea in the world for what should be done, and also the best person in the world to actually do that research? So that was the state of things in Castleman's, and we said, okay, we have to create a new way of doing research. We call it the collaborative network approach, and this is getting away from hoping the right application is submitted by the right researcher at the right time. So first, we spent a lot of time building and engaging a community of physicians, researchers, patients, even researchers from related conditions. Then we turned to the entire community to ask what research questions are important to you that can make an impact for patients. And we use the entire community worth of ideas and then hone in and focus in and prioritize the studies that need to be done. We go out and we recruit the best researchers to do it. Then we raise money. Then we procure samples. Then we perform the studies. And we ask the question, are there drugs that are already FDA approved that can maybe be useful based on what we're finding out in the lab? We disseminate knowledge. It makes our community faster and better at crowdsourcing, and we keep the cycle moving forward. So I've shared this slide at other rare disease events, and oftentimes people will say, well, how do you crowdsource and prioritize research? Give me some details. How do you guys do it through the CDCN? So I'm going to focus in on our most recent iteration, what we call AIM 2020, or the all-in movement. This was something that we launched earlier this calendar year in partnership with the Chan Zuckerberg Initiative. We actually been working with CZI for much of the last couple of years to create the infrastructure to make this more seamless and simple. It's something we've been doing for 7 years, but really this past year, we really doubled down on it. And that's where we built a platform at CDCN Connect, where patients, physicians, and researchers could actually submit their ideas over the course of a month for what research should be done. Then we organized and we lumped and we split questions, and then we had our community then vote on those research ideas. So then we had a rank ordered list that we could take to our scientific advisory board for their review, prioritization. Right now we've got it down to our top 20 list, and from that we'll select a 1 through 20. We'll rank order it, and then we'll start recruiting researchers that are the best people in the world at doing study 1, 2, 3, 4, and so on. So just some of the numbers over the last 6 months, we had 48 individuals submit ideas for research. So again, rather than having 3 researchers submit their 3 proposals, we're getting a lot more ideas, a breadth of ideas from individuals like patients, physicians, scientists that don't necessarily have a lab to do the work, but they can contribute priorities that are important to them. And we got a total of 155 ideas submitted, 48 individuals voted. We were able to lump the ideas into about 10 different broad categories. And then we got over 1,000 votes from those 48 voters. We could prioritize what are the categories of studies. So we didn't have categories ahead of time. We got ideas from the community, then we were able to lump them into certain categories. And then we asked the community to prioritize the larger categories. And maybe not surprisingly, but treatment and management came out number 1. But there were a number of questions that were not on our research agenda that had not been brought up previously, particularly related to side effects. There are some drugs that are really effective in Castleman disease. And as a scientist, I spend so much of my effort thinking about, does it work? Can it save lives? But our community reminded us that, well, what are the we need to be studying what are the long-term side effects? It may save my life now, but what does this mean for my future? What does it mean for my reproductive abilities? These are questions that I have to admit, even though I'm a patient, sometimes I lose sight of because I'm so focused on extending lives and finding drugs that could be life-saving. So from all of these different studies, it was our study ideas, 155. We've now got it down to our top 20. Like I said, we're going to turn into a specific 1 through 20 list over the next few weeks. So this is very much linked to an overall philosophy and lesson that I learned from my journey. And that's that I learned that hope cannot be a passive concept. We can't just hope that some researcher somewhere is going to apply for the right project at the right time that's going to make progress. Hope is a choice and a force. Hoping for something takes more than casting out a wish to the universe and waiting for it to occur. Hope should inspire action. And when it doesn't inspire action in medicine and science, that hope can become a reality beyond your wildest dreams. And I certainly am living proof that when you turn your hope into action, when you're hopeful, but you don't let that hope stop you from taking action, but you turn that hope into action, that you really can achieve that which you're hoping for. So the next lesson that I wanted to focus in on is the importance of joining stakeholders together. And specifically, I mentioned the importance of formal collaborations, where it's not just we collaborate as a community, but we collaborate in this way, achieving this particular goal. And so the first one I want to highlight is something called the SPEED 2 study. And this is something we worked on for several years. That we've got a paper coming out in the next few weeks. So I thought it'd be a nice study to highlight. First off, we spent 2 years reaching out to doctors all over the world, trying to understand how many doctors had blood samples in their freezers all over the world from Castleman's patients. Amazingly, we found that at 7 institutions, there were 356 samples that we could use for our study, locked in freezers for no one to ever touch, that probably would have been locked away for decades more. But we were able to unlock these samples by reaching out and saying, what blood samples do you have? So we got these 356 samples along with complete data sets on all of these blood samples. Next, the CDCN coordinated, Janssen Pharmaceuticals funded, and SomaLogic measured 1,300 proteins in the blood of those 356 samples. So really to quantify and understand what's happening, what's the immune system doing, and what's happening in the blood of Castleman's patients. And amazingly, a company called Medidata contributed pro bono bioinformatic analyses. So all of this work cost nothing. It took years of organization and lots of discussion. But what probably cost in total over $500,000, maybe closer to $1 million of work, cost nothing to the community, thanks to really incredible partners that joined together after, like I said, years worth of work. And we've made some really exciting discoveries. So the first is that now we can do a blood test we found that 7 of those 1,300 proteins, if you measure just those 7 proteins, you can predict with very high accuracy whether someone's likely to benefit from siltuximab, which is the only FDA-approved drug, or not likely to benefit. So if you just give everyone siltuximab, about 1/3 of patients will benefit. But if you run this test ahead of time, patients that have over a certain level are more like over 60% likely. So it can double your odds that you're likely to benefit. And on the other side, if you don't achieve this 7 protein test score, you're very unlikely to benefit from this one approved drug, which is important for a disease like Castleman disease, where the clock is ticking. Patients like me can be in the ICU within days of symptoms starting, and we don't have weeks or months to wait to see if the drug will work. But then the question you might say is, well, that's great if you can figure out who's going to benefit from the drug. Well, what do you do for people who aren't going to benefit from siltuximab? It's the only FDA-approved drug. What do we do for the rest of those patients? And so using that same 1,300 protein test, we were able to identify an aspect of the immune system that seems to be activated in patients that don't benefit from IL-6 blockade. And it's actually very much linked to interleukin-6. This particular protein I mentioned earlier, it's actually a part of this pathway or communication line that interleukin-6 signals through. And so we've discovered a new therapeutic target called JAK1 and JAK2 that we think looks really promising. So a new way to treat the disease based on this work. We did further what are called orthogonal studies, so looking at the same question from a different angle, where we, instead of looking in the blood, we looked in the lymph node tissue, and we confirmed that, in fact, the thing that we found that seemed to be up in the blood was, in fact, up in the lymph node tissue. And then, importantly, we went to the lab and we tested an inhibitor of that thing on cells from Castleman's patients, and we found that some of the abnormalities we find in Castleman's patients when they're in a dish could be fixed. They could be corrected when this drug was given to them. And so these sorts of steps, high level discovery, validating it in the lab, in tissue, and then validating it in what's called a functional experiment. These are the kinds of steps that lead you to believe that a drug might be a promising new approach. So I wanted to highlight that as one example that's coming down the pipeline. But a really important note that I also wanted to share is that this sort of research is really hard to do unless all of your samples that you get are very deeply linked with the clinical experience of the patient. So if I have a blood sample and I just know that it's a Castleman's patient, that is really hard to know how to interpret data from that sample. But if I know it's a Castleman's patient who at the time was on a particular drug, had just been hospitalized for 7 months before the blood sample was given, is about to be hospitalized again, all of their comorbidities, all the drugs they're on, that sample becomes 1,000 times more valuable. And we can learn so much more from that sample. And in order to do that, we launched what's called the Accelerate Natural History Registry to get that sort of extensive data. It's an international registry with a novel design, again, involving multiple stakeholders. Use of pharmaceuticals, UPenn, the Castleman Disease Collaborative Network, Janssen, and the EMA. I came up with this new approach. You can go to the paper at the bottom of the screen where we really get into it in depth. But the bottom line is that patients can enroll themselves directly online from anywhere in the world. Once they enroll themselves, we will then reach out to their hospital, their doctors, and we'll get all those medical records to the University of Pennsylvania. And then we'll enter line by line through thousands of pages of medical records, all of the data from that medical record. So we get the complete history into this database. Of course, we get blood samples and lymph node tissue when we can, and we correlate those clinical samples with clinical data. We now have over 400 patients. We're closing in on 500 patients. Our goal was 500 patients over 5 years. We're almost at 5 years, and I think that we're looking like we're going to achieve our goal. There are about 3,000 data points collected per patient. This is just a distribution of our patients across the US, Europe, and then this is a listing of all the studies utilizing data from Accelerate. And so literally over 15 studies right now have either been published or are in progress utilizing data from the Accelerate Natural History Registry. It really serves as a foundation for our scientific progress. So those are a couple of collaborations that have been just really transformative for us. Now I want to shift towards drug repurposing. As I mentioned earlier, I say drug repurposing is the fastest path. It's not the only path, but it certainly is the fastest path. And I think you'll agree with me as I go through a couple examples. So the steps that I like to think about when I think about drug repurposing, and a lot of people think about it in different ways, or maybe I'll take a step back. So drug repurposing is the concept of taking a drug that was originally developed for another purpose and utilizing it in a new way. So sirolimus was developed for kidney transplantation. It is saving my life for Castleman disease. Tocilizumab was developed for Castleman disease. It is saving patients' lives with COVID. This is drug repurposing at its heart. And so when I think about how do you do drug repurposing, the first step is to identify disease susceptibilities. I mentioned earlier JAK1 and JAK2. Laboratory research that we did identified that those two parts of the immune system seem to be really important. So that's maybe a disease susceptibility. We often use the fancy term therapeutic target, but really it's just saying something about that disease that we think that if we inhibit it, that we might actually improve the disease, or maybe we need to increase it and that might improve the disease. But what's a therapeutic target? The next question is, are there drugs that I can identify that already hit those targets? So maybe it's a drug that's already FDA approved, or it's a drug that's on a shelf at a pharmaceutical company, but are there drugs that can hit the target? And then third, can you do laboratory testing to confirm that if you use those drugs on cells from Castleman's patients or on some sort of a model system of the disease, does it seem to work? Does that patient or that cell line seem to have a positive response? And then what's really important is systematically tracking if the drug is used off-label, and off-label is the term to describe when a drug is used for a use that it's not approved for. So when I get sirolimus written for me, the rapamycin or sirolimus, the label is that it's only approved for kidney transplantation and LAM. And so if you're using it for something other than those two diseases, then it's an off-label prescription. There's a lot of off-label prescriptions. In fact, over 1/4 of all prescriptions in the United States are off-label. They're for things that drug is not approved for. So it's something that's done often, but it's rarely systematically tracked. And so we use the Accelerate Natural History Registry to track when drugs are used off-label for Castleman disease to figure out, do they actually work or not? And then finally, that's when you really need to move it into actual clinical trials to systematically assess, does the drug work when you look at it rigorously? So I briefly mentioned earlier my path to finding that a particular part of my immune system called the mTOR signaling pathway was highly activated in my samples. I performed experiments in the lab where we were able to confirm, in fact, this pathway or communication line was turned on in my own samples. And then I began testing this drug, sirolimus, on myself. Subsequently, we've studied this particular communication line in other Castleman disease patients and shared it through medical journal articles. I also mentioned that the Accelerate Natural History Registry has served as this wonderful resource for collecting data on when it works, when it doesn't work, and why it might work or why it may not work. And then based on the early promise we saw in the natural history study, we then launched a clinical trial called the RAPID clinical trial. And that was thanks to some federal funding. And it's important to note that a drug like sirolimus, it's already generic. And so it is difficult to motivate or incentivize companies that make generics to fund research like this. And so we had to get funding from the federal government or from another foundation or another source if we we're going to launch a trial. Thought I'd share a couple pictures. This is a picture of Joey. He showed up at the Children's Hospital of Philadelphia right next door to our lab a couple of years ago and was really, really sick. And he wasn't responding to first or second line therapy. And when he was given sirolimus, he had a really positive response. He was one of the first patients after me to receive sirolimus, and he's been doing really well for the last couple of years. Also want to highlight Deepa. I think Deepa was actually maybe the second or the third patient ever to get sirolimus. She lives in Australia, and she was very ill, was relapsing on the only drug that's approved for Castleman's, and had a really positive response. It's a picture of Deepa doing really well. Unfortunately, for every couple of patients like Joey and Deepa, there are also several patients that don't benefit from either the one approved drug, siltuximab, or the drug that's working for me, sirolimus. This is a picture of Lisa and Sergio, two young patients that failed to respond to any of the drugs available and unfortunately passed away because of their Castleman disease. And so highlighting for us just how much work we still have to do to continue to identify drugs for Lisa and Sergio. And we've been working on that. As I mentioned, we were able to identify this JAK1, JAK2 recently and think that it's an important direction. So based on that discovery in our lab, this young patient, Kayla, was hospitalized for most of 2020, spent about 7 or 8 months hospitalized in critical condition due to her Castleman disease. Nothing was working, even chemotherapy wasn't working. But based on what we were doing in the lab, we had just finished an experiment a couple of weeks before, and I had a conversation with this young patient's doctor and said, well, if you're out of options, we just got data back on this experiment a couple of weeks ago with this JAK inhibitor. You could consider trying a JAK inhibitor. And Kayla had a really, really positive response. This is just an early indication of how long she's been out of the hospital. I think it's now been another about 7 or 8 weeks since we put this graph together. So it's now been, I think, close to 40 weeks that she's been out of the hospital since starting on this drug, ruxolitinib. And you can see compared to her previous hospitalizations, really, really notable improvement. We also just a couple of weeks ago published in The Lancet the first ever reported case of JAK1/2 inhibitor, another drug called baricitinib, and how it helped a patient in Vancouver. And so I wanted to highlight these because it shows just how quick you can go from a discovery in the lab. Now, of course, it was years of work to get that SPEED2 study launched. Literally years of banging on people's doors to get those samples, taken out of the freezer so that we could study them. But years of work to, once we made the discovery, a matter of weeks before patients were being treated with JAK inhibitors in Vancouver, in Paris, in Chicago, in Philadelphia. It's really, really pretty incredible. So I mentioned we've described this recently. Now we're looking to potentially launch a trial of JAK inhibitors. So following this sort of approach and the success that we've had in Castleman disease, we felt it was important to systematically track drugs for COVID-19 and to identify drugs that look very promising from the data. And really excitingly, I had the opportunity to serve on the working group that selects drugs for the largest trial that's ever been launched for COVID here in the US. It's called Active-6. And we utilize data from the Corona Project to select the drugs for Active-6, and that study is now open. So hopefully they will be effective in the trial. So just in the next couple of minutes as I close up, I want to highlight some frameworks that you all can think about as you think about potentially looking at drug repurposing for your given disease. First off, the first step is to identify and validate potential candidates, drugs that might be already FDA approved, that might be able to be used in a new way. One way to think about it is the way that we do it, this target first, where we try to understand what's going on in human cells. And then we say, well, what drugs are approved that can fix that thing that we find wrong? You can also use published literature. Groups like HELAX use published literature. You can also do high throughput drug screens. NCAS does a lot of high throughput drug screens where you're literally mixing drug with cells and you're looking to see if there are combinations that maybe you didn't even anticipate, that look promising. You always want to validate that, and then you want to start tracking when drugs are given to humans. You don't want a patient to be given sirolimus or whatever it may be, and not learn from it. So that's why it's so important to have these registries in place so that you can get data on how well those drugs are working. And then if drugs look promising, you got to move them forward to clinical trials with the goal of either getting FDA approval or adoption in clinical practice. These drugs don't have to be FDA approved for the new use. They need to be FDA approved for something to be able to know that they're safe and can be used. But they can oftentimes, as I mentioned, about a quarter of all drugs are not approved, or all prescriptions are not for an approved use of that drug. There are a lot of ways to collect this sort of data from patients, from physicians, the published literature, electronic medical records, and also insurance claims data can provide a lot of this information. I also wanted to boil this down into just what I think is, or hopefully is, a simple framework. And that's that we oftentimes think about a drug as being a drug for a disease, but drugs are not developed for diseases. Drug like sirolimus, though it was developed for kidney transplantation, it doesn't work by fixing or preventing rejection of kidneys. It works by inhibiting something. It works on a target, and we've learned that inhibiting that target is good for preventing a disease. Same thing with tocilizumab. It is not fixing Castleman disease, it's blocking interleukin 6, and interleukin 6 is important to Castleman's. And so you can imagine when that thing that's important to Castleman's is also important to other diseases like rheumatoid arthritis or COVID, then that drug now becomes helpful for other diseases. And interestingly, a lot of these diseases have multiple targets. There's many things that if you block them or if you increase them could actually be helpful in fixing that given disease. And so other drugs might be possible, and even diseases that you hadn't even thought about before that might benefit from a drug because you don't even know all the ways that drug may work. So thanks to this kind of approach and the work that we've done, we've made a lot of progress in the last 9 years for idiopathic multicentric Castleman disease. This slide is my attempt to start out by showing where we were in 2012 in white, and then I'm going to superimpose in blue where we are today. So from about $10,000 a year being spent on translational research to over the last 9 years, raising and spending about $1.8 million on research, and then an additional $11.1 million being spent on IMCD research from outside organizations. So we find something early that's interesting and promising, and then an outside organization will invest significantly more money into validating and better understanding that. In fact, over $13 million has gone to other organizations based on some of the preliminary work and other diseases based on some of the preliminary work that we've done. That's CZI funding. That's also the Parker Institute for Cancer Immunotherapy that we've been able to spur. The NIH funded our team to study Castleman's for the first time ever. We didn't have a registry. Now we have over 1,200 patients in a registry. And 400 of those patients in our natural history study. There were multiple organizations. Now we have one unified collaborative network. And I don't have too much time to dig into this deeply, but in a lot of rare diseases, there's a real proliferation of nonprofits, and every organization within that particular rare disease is competing for progress and competing for finite resources. And I think that in Castleman's, we're very fortunate that the two organizations that existed before the CDCN were very open to joining together into one unified collaborative network. All three of us work together under one umbrella, one organization. And I think that this is partly why we've been successful. We have a unified research agenda. We have a biobank that samples go to. Many questions still remain, though progress has been made. We have a diagnostic criteria, an ICD-10 code. We've published treatment guidelines on how do you treat the disease that didn't exist. We have blood tests that can predict whether someone's going to benefit or not. There's an FDA-approved drug, cetuximab, that works really well for about a third of patients. And then we've got 2 more drugs in development with 3 more that we've identified that are currently under consideration. And importantly, all drugs being tracked in one database. So bringing it all together, if I were to try to put this all into one slide, I think it's essential that you engage your entire community. We use CDCN Connect to do this. You use your scientific advisory board to establish ICD codes, diagnostic and treatment guidelines. You crowdsource from your community the various research ideas. We use AIM for that. And all 3 of these fall into that bucket of patient engagement and crowdsourcing that I mentioned earlier. Moving forward, you have to obtain biospecimens through a biobank, clinical data through a natural history registry, and perform large studies to identify susceptibilities within your disease. This all falls into the sort of multi-stakeholder collaboration bucket. And finally, as you understand susceptibilities, then you want to confirm those targets in the lab. And you then want to assess whether they actually work when they're given off-label or in clinical trials like our Accelerate study and RAPID, really focused on drug repurposing. And then the last piece of this, in our opinion, something that we're really passionate about is making sure that we share our learnings across the rare disease community. It's not good enough to find a drug that's saving Castleman's patients or saving lots of Castleman's patients. If we do that, I feel a real responsibility that we need to share how we did it and what we did to achieve that so that other groups can follow a similar roadmap or a similar path. And actually, we've launched a project called the Roadmap Project, again, along with the Chan Zuckerberg Initiative, to literally map out what are the steps, what is the path for different groups to get from no drugs to drugs that can be repurposed. And so be on the lookout for progress. And our goal is to get from there being 2,500 drugs approved for about 2,500 diseases to a state where those 2,500 drugs are used for many of the other diseases that don't have any FDA-approved drugs in partnership with CZI and also their Cured Drug Repurposing Collaboratory that I have the honor to co-direct. And one of the reasons I wrote Chasing My Cure was to share this example of an existing drug saving my life that no one had ever thought to try. So I'm just going to close with a few lessons, and then I'm eager for Cara and I to spend some time chatting. So the first is that hope can have many different forms. I talked about how we went from hoping that a drug or that a researcher would find a drug to really turning our hope into action and to really driving forward this AIM project where we're literally asking for ideas from the whole community instead of just hoping that someone's going to have a good idea. The second is that multi-stakeholder collaboration is key. The speed study, if that hadn't been done, if we hadn't literally spent years organizing and coordinating that, we would not have made the discovery about JAK1 and JAK2, and Kayla would likely not be alive today. And it's these sorts of long timelines. It was 5 years of work before this drug could save Kayla's life, but it's work that had to be done, and it's really critical. And how many more lives will we save because of it? The third is that solutions may be hiding in plain sight. As I shared, the drug that saved my life was at my neighborhood CVS for years while I was in and out of the ICU, but no one had thought to try it, and no one knew that it might be helpful for me. How many drugs are just sitting there in your neighborhood CVS that we have to unlock? The fourth is that we have to share our roadmaps with one another. This can't be, like I said earlier, it's not good enough for us to make a discovery that's going to save Castleman's patients' lives. We've got to share those discoveries, those paths, these plans with other rare diseases, because we're all in this together. I have a really talented PhD student named Anya who's leading up this effort to track and create roadmaps for drug repurposing. So please email Anya at castlemannetwork.org if you're interested in collaborating with us and sharing your experiences, what you would find useful that we could share as well. And finally, it takes really all of us working together. As I shared earlier, my book's called Chasing My Cure, but it really should have been called Chasing Our Cures, because it really has been such a team effort. And please do send me an email if you don't have a copy and you'd like one. I'd be thrilled to mail one to you. Of course, it's available everywhere books are sold and on Amazon. But if you'd like one, I'd be happy to mail one to you. And I just have to thank my amazing team and all of our partners who make all this progress possible. So, Cara, looking forward to digging into some questions.
David, thank you so much for sharing your amazing journey. I have a lot of thoughts and questions already, and there are some coming in from the audience as well. But before we jump in, I just want to share a couple quick reminders to the audience. We would like you to please drop your questions in the questions box to the left of your session video. Questions in the chat are actually not visible to the speakers, but you can also upvote questions from other attendees to help the speaker prioritize what they will address. So let's just jump in here. The pandemic has raised more awareness in repurposing. Is there an opportunity to expand that conversation?
I think so. I think that the fact that tens of thousands of lives have been saved with dexamethasone, which has been around forever, with baricitinib, with tocilizumab, these are drugs that have been around for decades, and they're saving COVID patients' lives. I think this is just highlighting the potential for drug repurposing, and I think it's important that we really leverage this opportunity.
Great. Yeah, I totally agree. Well, thinking about the past year and the evolution of the conversation, equity and access, are there lessons from that we can take away?
Absolutely. I think that so many of us have really started to take an extra hard look in the mirror at the way that we do research and the way that we provide medical care for everyone. And I think that the rare disease space is an example where we know that individuals that come from variety of backgrounds will have a variety of experiences with their disease, and that's not the way that it should be. We have to strive towards a future where everyone, regardless of what your background is, or whether you happen to be a medical student while you get sick or whatever it may be, we all need to have a uniform experience. And I think that the rare disease community, I think, has been spending the time reflecting on this, but we've got a long ways to go. And I think first we got to realize that there's work to be done. But I do think that, as was mentioned earlier during the talk, that things like e-consults and the ability to visit patients at their home by virtually, hopefully this can help with some of the inequity that exists.
Yeah, we've definitely heard the same. We have actually some questions about international collaborations. Now, what is needed to advance international collaborations and leveling the playing field? But also, we had some questions about multi-center clinical trials and if people would be interested in having a center in places like China if they're not able to get to the marketing approval immediately. So are there any factors that they would consider besides profit or marketing size when making those choices?
Yeah, these are great questions. Rare diseases by definition, we're all rare, of course. And so what that means is that we can't do sufficient work at one institution. We don't have big enough sample sizes. Even if every patient, is going to one place, we've got to collaborate. And I think that should be a principle across all of medicine. It's not. But in rare diseases, we have to recognize that none of us can do it alone. We've got to collaborate. And we really likely can't do it within even just one country. It really does, I think, require international collaborations. And so that one slide I shared with the 7 different institutions, we literally, I guess we figuratively knocked on every door, but we emailed hundreds of doctors around the world to get a hold of these samples. And thankfully, the 7 or 8 institutions actually, opened up their freezers and gave us the samples, but we couldn't have done it if we just said we were going to stay within the US. So I really do encourage international collaboration. It's not always as easy and it requires more paperwork. And like I said, that study, from the time we started working on it until now, it's been about 5 years that study has been going on. And, we all wish that we could shorten those timelines. And I promise you, we tried to. There was a lot of work to make shorter timelines than that. But it's important to do because, like I said, there's patients that are going to benefit when you do it. And then the question around clinical trials, first, multi-site clinical trials, I think, are always going to be better than single-site clinical trials. The more diversity you have of patients, the more likely that you're finding in your clinical trial is going to be replicable and actually, reflect clinical experience. I think that's always a good thing. I think that we've had some challenges collaborating with some centers in China and the ability to have data and samples leave and come back and forth. But that's, I don't think, a uniform experience. I think there are ways and there are examples where it can be done a little bit more frictionless.
Great, good information. We have a question about the pipeline, and you spoke a little bit to this as well already. I'm going to combine a couple of them. One asks about how do you fasten the pipeline, and also, is there AI? Can that play a role in making this faster for all of us?
Yeah, I think that's a great question. I think the first step to speeding up the pipeline is to creating a pipeline. And by that, I don't just mean the things in the pipeline like sirolimus and Jakafi. I mean literally having a place that you can put all of your studies. If you go to cdcn.org/research, you will find our pipeline. It's literally all of the research that we're either funding or is happening anywhere in the world for Castleman disease. That to me feels like a critical step. You can't really track what's going on unless you have it all in one place. And so first off, create a pipeline that's truly tracking everything. So every researcher in the world, patient knows what's going on. And then as far as how do you speed it up, I think that it requires dedicated effort. And, in the rare disease space, we often, at least historically, would think of a rare disease organization as being a funder. That's what we do. We fund research. And then we, help to cheerlead and champion when that research turns into a breakthrough. We help with enrollment in trials. But I think there's been a real shift over the years. And it's thanks to some amazing leaders in the rare disease space like Josh Summer, Emily Kramer-Golonkov, and so many others that I don't even have time to list today. But that sort of change is where they've said, we're not just going to fund and we're not just going to help to enroll in trials. We're actually going to drive and shepherd rare disease research. And I actually like the word shepherd, and I like the analogy of a shepherd because researchers are often like a herd of sheep, I guess, in that they're kind of all going in their own direction. They're usually moving way too slowly, and you need to focus them. And you really need a shepherd to keep this stuff moving forward and to make sure that, when one of your studies is slowing down, when one of those sheep is at the back of the pack, that you can really start pushing it forward. Researchers don't always love the idea of research organizations being shepherds because, we like, to do things on our own time, and we've got a lot of work going on. But as a research organization and as a funder, you have the opportunity to build timelines into your grants and milestones so that they don't get paid unless they hit their milestone. You also have ways, to highlight the groups that are at the front of the pack and to, make sure that you're encouraging and supporting those that are at the back of the pack. And so I think that build a pipeline and don't be afraid to be a shepherd. And you can blame it on me. You can reach out to a researcher and say, you know what, I wasn't going to follow up with you the 5th time, but you know what, David Faigenbaum said that, we should be shepherds. And I'm sorry, but here's my 5th email. But truly, I really do think that sending those emails, checking in, shepherding is really important. And you shouldn't be hesitant to do it.
And along those same lines, for multi-collaborators, how do you get them to work together?
It's really hard. It's really hard. I mean, I think that, a lot of times grants will require to have multiple groups involved in it. And that's such a great idea theoretically. But in practice, what it often encourages is people to agree to work together on paper to get the grant. And then the money gets split up and then they just, work on their own in silos afterwards. So it's really hard to create these sort of fundamental collaborations. And the best way, in my opinion, to do that is when you have various members of your collaboration that are really, really good at one thing, but they can't actually move forward without another piece of the puzzle. So that Speed 2 collaboration is a great example of that. SomaLogic is the best company in the world to quantify proteins, but they can't quantify proteins without samples. The CDCN, in my opinion, is the best organization in the world to organize a study like that and to get samples out of freezers. So we can do that. Janssen Pharmaceuticals has the resources to actually fund a study like that. So they're at the table. And it's these sorts of things. Oh, and then importantly, Medidata has this incredible bioinformatic capability that they've deployed towards this project. And so, none of the players could have done this on their own. It really requires this collaboration. And so if you can establish collaborations up front where it's clear, you're here because you're really good at this. I'm not good at that. I need your help for that. I think that's really, really important. Formalizing it into contracts is actually more important than maybe I used to think it is. But you want to formalize it in writing. You want to make sure that philosophically you're all on the same page. And then I think it's also important, just thinking about philosophy and mission, is that you want to remind everyone that it's always all about the patient. I mean, I think that a lot of us in medicine can lose sight of things and we focus on one thing that's wrong or the right thing. But gosh, as a patient organization, you have the opportunity to include your child on the call with the researcher, to bring your whole family and your whole team in to be a part of meeting these different researchers and to keep reminding us that it's not about the publication, it's not about the number of citations, it's all about the patients. And we in the rare disease community have the ability to remind the scientific community that.
That's really great. Thank you. That's a lot of great tips for people going forward and setting up these collaborations. So actually, I think we're coming to the end of our question and answer period, but I think it'd be great to end on understanding how the patient groups and researchers can be a great partner together. And maybe what are some of the best practices that you can share with us about how to have those interactions?
Yeah, it's a really good question. I think that maybe my number one lesson would be work together early and often. Don't make it something where we're going to have a patient group and researchers get together once a year. I mean, once a year is great, but get together in person once a year when it's safe, but also email frequently, set up phone calls. You want to make sure that it's something that's symbiotic, where it's not just that you as the research or you as the group are just always calling the researcher to get updates. But think about ways that you can give updates on the patient community. What's an anecdotal report of a patient that maybe got a treatment that maybe looks like they're getting better, that might be interesting to those researchers. And so I think about it as early and often, lots of engagement. It's a partnership, and none of us can do this alone. I mean, no one in the rare disease community can make progress on our own. It absolutely requires working together with others. And so don't lose sight of that. And that includes you as the rare disease group. No one in the community can do this without you. And so don't lose sight of your value, your importance, and don't forget about how valuable being a shepherd is. If someone else is losing sight of timelines and progress, you can rein them in. You can remind them of the importance and the impact of their work. So I think those would probably be the best practices. And the only other one I would add in is just always reminding the research community of the impact and the importance of advancing research for that disease.
Thank you. This has been a really valuable conversation. Really appreciate your time and all your thoughts and insights today. And we want to thank you to the audience for some really great questions and continuing this discussion. I know we didn't get to all of the questions, and we'll do our best to follow up with those offline after we wrap up as well. But before you head to the breakouts, please take a few moments to respond to the polls that you'll be seeing in Launch Now. We appreciate all your feedback on the sessions and want to assure that we're able to provide great content and programming for the rare disease community. You can also share your feedback at events@globalgenes.org, either now or in the future, and be sure to take some time to explore the events page. Visit the posters and build your itinerary for the remainder of the week. Plus, visit the Achievements and Badges tabs to learn more about how you could win a pass to a future Global Genes program. So thank you all so much for joining us. We hope to see you in person sometime soon.
Thanks so much, Cara.
Thank you.